Today's presenter is John M. Lehman, PhD, and professor in pathology and laboratory medicine. Dr. Lehman has given several previous MHIG presentations, and so here is Dr. Lehman to speak on poliomyelitis, history, conquest, and cost analysis. Dr. Lehman.
Okay, thank you for that very nice introduction, Melissa. Today I'm going to talk about poliomyelitis and sort of a little bit about the history, a little bit about its pathogenesis, and kind of the vaccine development. And one of the reasons that I was very interested in doing this is, even though I've never really worked with polio, I have actually worked with a virus very similar in mouse called Mengo virus, but never with human polio. But I spent my early years at the Wistar Institute of Anatomy and Biology in Philadelphia. At that time, the director of the institute was a gentleman named Hilary Koprowski, who you'll hear about. And he was very much involved in the early work and development of the polio vaccine. So what I thought I'd do is start here and talk a little bit about the history of poliomyelitis. And actually, it's a disease that dates back to antiquity. Now, whether this individual, who is actually a high priest in the religion of Egypt, had poliomyelitis or not, or some other affliction, you can see that right leg is much withered and really looks very much similar to modern cases of poliomyelitis. So, and then if we track back in other times of history, we also see paintings and so forth fairly similar. So polio has been around for a very, very long time. This is a little history of kind of what I'm going to talk about today. You'll see this again. So back in 1796, Edward Jenner, and we're coupling sort of, this is a virus, and it's also very important from the point of view of cure and ablation prevention to development of a vaccine. So the first vaccine that was ever developed was actually Edward Jenner, who recognized, by actually listening to the local people about cowpox vaccinia, and people that in a sense got cowpox never developed smallpox, which in those days was, and even today, is a very debilitating disease. So this was the first use of a vaccine. And then almost 100 years later, at the time of the discovery of microorganisms by Louis Pasteur and also Robert Koch, Pasteur developed the rabies vaccine. And maybe at some point in time I can give you kind of a historical perspective, because I know quite a bit about the rabies vaccine also. But then it wasn't until a few years later, in 1892, that two individuals recognized an agent that was filterable. And they were studying tobacco mosaic disease, which is actually apropos for North Carolina, who is a big tobacco, or used to be a big tobacco growing state. And they basically suggested that this was a filterable agent, and they coined the term virus. One was a Russian, and one was a Danish individual. In 1908, which is only a few years later, not even 10 years later, Karl Landsteiner. And for those of you that follow medical history, Landsteiner is the discoverer of the ABO human blood cell types. At the time he made that discovery, he was at the Rockefeller in New York City. At this time, he was in Germany, that's his home country, and he was a physician, and a child came in with poliomyelitis and developed full bulbar and wound up, and encephalitis, and wound up dying. And what he did was he took some of the material at autopsy, ground it up, and he fed it to two old world monkeys that were in the laboratory. And about a week to 2 weeks later, both of these animals came down with a disease very, very similar to what the child had died of, poliomyelitis. In fact, the pathology was very similar. In a sense, he satisfied Koch's postulates for the recognition of a microorganism causing a particular disease, and that was in 1908. In 1916, actually in New York City, but actually a few years before in Vermont, there was some major outbreaks of poliomyelitis in the community. So much so that New York City almost came to a complete stop in 1916, 1915. In fact, hospitals basically shut down, and I'll show you a little poster from that era from another part of the country, because they had no idea. Even though Landsteiner recognized it was a virus, they had no idea from a public health point of view really what was going on. And then when the international organization began to keep records of polio incidents, it was clear that the developing and prospering countries were the ones that were showing the highest incidence, and I'll show you some data in a minute. So that was 1916. And as a result of work that went on over the next 20 years, two individuals, one in New York at NYU and the other one in Philadelphia at Temple, actually developed the first polio vaccine and actually did some field trials. It was a disaster. In fact, when they analyzed the data, which really wasn't well controlled, it turned out that they were actually giving the children that received the vaccine polio. So this put a whole stop on polio vaccine until we sort of generated more data. In 1944 to 47, three individuals, Bodin, Howe, and Morgan, demonstrated that the poliomyelitis was caused by three different viruses, type 1, type 2, and type 3. And they did these studies based on what they knew about immunology and recognizing different immune sera from these three groups. And they at the time, the main laboratory animal, or the main study, was done on animals and monkeys. And old world monkeys were primarily used, as well as some new world monkeys, which we'll mention in a minute. And in 1946, the Letterly Laboratory in New York, outside of New York City, began working on a polio vaccine. So that's almost 10, 15 years after that fiasco that occurred in 1935-36. Then a major, and that's why I have it in red here, John Paul, and I have his book. He was a faculty member at Yale University, and he's actually written a very nice history, which I'll show you reference at the end. The library here has the book, and it's sitting over there. It's very difficult to find. We happen to have a copy. And he put together a very exhaustive history of polio. And this book, I believe, was published about 1970, 1971. And it's one of the classics of medical history that Yale published in this regard. Now, Paul was also an active researcher, and he made a very interesting observation back in 1946. And he found that polio, by looking at we didn't have a World Health Organization at the time, but the individuals keeping international records were finding out that polio was prevalent in most developed countries like the United States and Western Europe, and was not occurring or was not being seen in underdeveloped countries or countries like India, China, and in Africa and also in South America. And he basically published a paper suggesting that something very unusual was going on here from a public health point of view, and we'll talk about that in a minute. 1948 was the foundation or beginning of the National Foundation for Infantile Paralysis, which is also known now as the March of Dimes. As a result of the development of vaccine, they changed the name to the March of Dimes, and most of what they do now is basically study genetic diseases. But that's an important milestone, and we'll mention that in a minute. And then probably along with the Landsteiner and then the recognition of the three types of poliovirus, a very major discovery was made in 1948. And that was made by John Enders, who was a virologist at Harvard. At the Children's Hospital, he ran the virology infectious disease lab there. And two residents, pediatric infectious disease, Dr. Weller and Dr. Robbins, who were working with him, made a very interesting observation, and that was they were able to culture poliomyelitis virus. Actually, they're working with the type 2 strain in cells other than neuron cells. They were able to culture them in a whole group of human cells, and we'll talk about that, why they were able to do it. And this basically was a major milestone in 1948. The three of them won the Nobel Prize in 1954. I might mention that the literature prize was Ernest Hemingway, and the prize in chemistry was given to Linus Pauling. And it's also rumored that Enders basically would refuse the prize if the two younger individuals were also not named. And in a sense, that gives you an idea. By the way, he was a multimillionaire. His father was a businessman, a banker in Connecticut. That's where he grew up. And it's purported that he inherited something like 19 million at the time. And so money didn't really, and by the way, the Nobel Prize wasn't very large in those days compared with today. But to him, finding the cause and understanding basic science was very important. And by the way, his background, he was initially, his main area of interest was English literature. And he started out as a professor in English literature and then decided he was going to go back, not to medical school, but he's a PhD, and went back to, I believe, Yale, and that's where he got his PhD, then went to Harvard. Now, I'm not going to spend a lot of time here because we're going to hear about this later. It's really 1947, the Letterly Laboratory started the polio virus work, and the two individuals involved in that are Harold Cox and Hurley Koprowski. And then obviously all of us know about Salk polio vaccine, and Jonas Salk began to work on this, and then another individual, Albert Sabin, who started to work on the live vaccine. And we'll spend some time later on talking about these individuals. And as you probably know, the Salk vaccine was approved for use in 1954, 1955, and really about 1961-62, the oral Sabin vaccine. And you'll hear more about that in a little bit. Okay, here's Carl Landsteiner. I got this picture from the John Paul book. A very distinguished looking man, and as I mentioned, he had a career that extended not only through 08 when he was in Germany, but also at the Rockefeller. Now, for those of you that, I gave a lecture last year on Paul de Kruif, an individual that was actually a card-carrying microbiologist, a graduate of Michigan, who worked at the Rockefeller, and the director of the Rockefeller Institute at that time was Simon Flexner, the first one. I think I mentioned a little bit about him. So Simon Flexner made major contributions to infectious disease. One of the areas that he made contributions to, and he also in some ways was a detriment, was poliovirus work. Because the Rockefeller kind of took over polio. Now if you look at number 4, the Institute was very successful in using a treatment for individuals that came down with a form of polio meningitis caused by poliovirus. And some people in Europe, but here, primarily at the Rockefeller, they got great success by actually infusing into the spinal column this horse gamma globulin and basically prevented the meningitis. And in fact, the Rockefeller and its affiliated hospitals were the main areas for people to come for treatment for polio meningitis. However, Simon Flexner made a major error when he started working. He chose as his model monkeys, which was the main model for everyone, including Landsteiner, but he used a New World monkey. He used the rhesus monkey. The rhesus monkey is one monkey that the natural transmission cannot occur, which is through the oral pharynx. He thought that transmission was actually due the respiratory canal, so that aerosols, this is how it was basically transmitted. And so for about 20 or 30 years, we had this whole problem with really how does someone become infected with poliovirus, and that along with the fact recognizing it was a virus. His route of entry was nasal, not the GI, and based on experiments that actually, if he read Landsteiner's paper carefully, that's how Landsteiner produced it in those two monkeys, was actually by feeding a gamish of the material taken at autopsy from that child. The other thing that he did was, Simon Flexner, he produced a strain of polio, MV. He also didn't believe that there were 3 strains. He believed there was only 1 strain. So that also went against the work of those 3 individuals who showed that there were 3 strains. This was actually produced in monkeys by intranasal injection, and there was then transmission to the central nervous system. But when the virus was extracted, not only did you get the virus particles out, but you also got a lot of neural protein. And that's a major, major problem which they ran into with the rabies vaccine, is whenever you make a vaccine and you have neural, you can get cross-reactivity. And so that was a major problem with his work. He had a major influence on polio research, most of it positive, but the small 2 or 3 instances I mentioned were kind of important because it set everything back. Because the Rockefeller at that time was the shining virology institute in the world. As I said, Carl Landsteiner went there. Tom Rivers was also a major individual. He was one of the most recognized virologists in this country for over about 30 or 40 years. And actually, many of the people, Albert Sabin actually trained at the Rockefeller before he went to the University of Cincinnati. And for those of you that were here last year for this talk, when I talked about Paul de Kruif, Sinclair Lewis wrote a book in collaboration with Paul de Kruif called Arrowsmith. And one of the individuals or characters was the director of the McGurk Institute, which was the Rockefeller, and that was DeWitt Tubbs. And he was not treated quite nicely in the book. Anyway, these are the 3 winners of the Nobel Prize in 1954, Enders in the middle, and Robbins and Weller. And I think Dr. Pori sent me an email. He made a comment that when he was training at Rochester, University of Rochester School of Medicine, the dean was Fred Weller, who had moved from Harvard to become the dean, and they told me a little bit about him. These were 3 elegant gentlemen, and I actually have a book that Tom Weller wrote basically about his hunt for viruses and the use of cell culture, and there's a very large portion in the book about the poliovirus. So let's talk about the pathogenesis. And most of these I've taken from lectures that I've given to medical students and graduate students, and it's actually taken from the Murray textbook that we use for the first-year medical students in virology here. So poliovirus is a member of the picornoviruses. Pico means small, and RNA, it's a small RNA virus. It also, and type 1, type 2, and type 3 are polio, but there are a whole bunch of other viruses. They're part of this group. If you look down to the next big group, it's rhinoviruses. These are the common cold, of which there's over 100. So in some ways, Simon Flexner wasn't far off. Many of the viruses associated with picorno are actually upper respiratory tract viruses. And so he was just misled, maybe because of some other studies. Okay, this is what the virus looks like, taken from a picture from the CDC. It's about 25 to 30 nanometers in diameter. It's an icosahedron structure. The enteroviruses, of which polio is part of, are very interesting. They're very resistant to pH ranges of 3 to 9. They're also resistant to detergents and mild sewage treatment, and we have to remember that also doesn't destroy them, and also heat doesn't. The genome is a messenger RNA. It's a single-stranded RNA that's around 7,400 to 800 base pairs long. And for those of us interested in virology, it's the best well-studied RNA virus around. My virus, SV40, is the best studied DNA virus. The viral RNA is also capable of infecting cells, although the efficiency was much lower. The virus replicates in the cytoplasm, and the interesting thing is when the virus is uncoated, and I'll show you this in a minute, the viral RNA becomes a messenger RNA. It goes right to the host ribosome and is translated into a very large protein, and this is how it begins to initiate its replication. And the viruses are cytolytic. And the important thing in here, many viruses that we deal with wind up playing tricks with the immune system, either suppressing it or as a result of the antibodies made, actually the antibodies react against the host. In this case, the lysis of neurons and other cells by the poliovirus is as a result of virus replication. This is the viral genome, 8,400 bases. On the right side is the polymerase, which is an RNA-dependent RNA polymerase, which when this protein is made is cleaved by one of those proteases there, and VP4, 3, 2, and 1 are the coat proteins. This is sort of, again, a schematic showing the virus enters the cell through a vesicle here, and the key is for cells to be infected, they have to have a specific receptor which matches a key protein on the virus coat. And cells that are infectable have that, and cells that are not do not have it. Okay? For instance, mouse cells cannot be infected with poliovirus unless you put that gene into the mouse cells from the human, and then they become infectable. And we don't have time to talk about that. So you make this, the RNA is transcribed, it makes a polyprotein, the polyprotein is cleaved, and the protein then makes the late proteins for the capsid. It then creates that polymerase, which then makes the viral DNA, RNA in the particle is a positive strand, a negative strand is made, and then off the negative strand a positive strand is made, and then you get encapsulation, and you get new virus particles out. And that's sort of the replication cycle, which usually occurs probably over about an 8 to 12 hour time period. Okay, how do we get infected with? And this is a key and an important point that took a while for individuals to decide. We're the carriers of the virus. Those of us that are infected pass major quantities of it out through the feces, so that anything that comes in contact with our hands or sewage or waste is a potential transmitter of the virus. And that was an extremely important key recognition. So hands, sewage, solid waste, landfills, water supply, shellfish. That's how it's transmitted. And those individuals that recover from the virus will secrete it for many, many, many months and maybe even years. I know that because when my wife was a technician at the Wistar, she worked on the poliovirus, and one of the things that they were doing was actually characterizing this from some of the vaccine trials. So those of us that get vaccinated with the live polio vaccine, the Sabin virus, will secrete lots of virus, and so you develop herd immunity. So it enters the intestinal mucosa. Oropharynx is because of the rhinoviruses. In absence of serum antibody, enterovirus spread by viremia to cells of receptor-bearing target tissue. And receptor-bearing is neurons. So one of the other key important findings in all of these studies was the fact that you get an initial infection, and most of that occurs in the lymphoid tissue of the Peyer's patches of the GI tract, and also the lymphoid of the oral pharynx. Virus gets into the circulation, and that virus then can be, when and if it's transmitted to the central nervous system, will infect the neurons. And one of the problems with neurons is they're not able to replicate themselves. So when you kill a neuron, which the poliovirus will do, its RNA, it's gone, it's finished. Okay, and if enough of them are replicated, you have a problem. And we know what the receptor is. The infected target tissue determines the subsequent disease. And viral, rather than immune, is the pathological effect. That's another important point. Now, secretory antibody response, the initial secretory antibody response is weak. But then later on, 7 to 14 days after the infection occurs, you get a full antibody response, and that's key. That recognition said, oh, one way we can really maybe solve this poliovirus is to develop a vaccine that the host will then respond to and produce enough antibodies so that when they come in contact with polio, they'll have a prior immune. And we knew type 1, type 2, and type 3 were the important ones. And the serum antibody blocks the spread. And as I mentioned earlier, the enterovirus is shed for long periods of time in the feces. And infection is often, and this is key, is asymptomatic. You'll see the data in a minute. So many of us can walk around with mild infections and actually go to work and do the various things and transmit it to other individuals. Okay, so this is a schematic from Murray. This is all of the picornoviruses, but we'll just focus on polio. It replicates in the GI tract, in the Peyer's patches. You get a primary viremia and also some secretory antibody. And if you have antibody there, it blocks it so it doesn't get to the secondary tissue, in this case the meninges and also the brain. Okay, and with the rhinoviruses and the other one, the Coxsackie, they're other target tissue. Schematic, virus, the first 5 to 6 days, the oropharynx, you get a viremia. Virus begins to appear in the stool about 6 days. These are some of the symptomology associated with this mild infection. It's a minor illness. But if you get the CNS invasion, it turns into a major, either meningitis or bulbar poliomyelitis, where it infects the cells that service the respiratory center as well as motor neurons to the arms and legs. And this is about when the antibody, 7 to 14 days, are occurring. So the question then was, if we can enhance and get people immunized, we would be all set. This is just showing the two vaccines, the inactivated, killed, and then the live polio vaccine. And the live polio vaccine is one of choice for many areas because you get secretory antibody that will protect the oropharynx as well as the GI tract. So nasal IgA and also duodenal IgA. Okay. Transmission. Fecal oral route. That's clear. Now, who's at risk? Young children are at risk for polio, but in most cases it's asymptomatic. And this goes back to Paul's observation. Why did we see such a problem in the developed countries like the United States and Western Europe and not in other areas? Okay, because we've done the epidemiological studies, polio is there. Primarily the reason was that the children in the underdeveloped countries were infected early. And most of the mothers breastfed, and what they had done was they had become immunized by polio, and they had passive antibodies which they transmitted to their children. And so for many months to years, they had this passive antibody which basically blocked that primary response, and then they could kick in their secondary response because the viremia was suppressed. Individuals that are infected, older children and adults, like in the United States, and I think the best example, you'll see him later, is Franklin Roosevelt. He became infected, I think he was 38 years old, 1921, and he developed paralytic polio as a result of never having seen it as a young child. This bit of information that Paul was able to see by his epidemiological studies and others was this is a possibility of either infecting individuals or getting passive antibody transfer through the colostrum, through the mother's milk, and getting kids immunized early, and that's been the key. It's widespread, basically more common in the summer, and modes of control now are actually the polio vaccine, both killed and live, and good hygiene. But the issue is probably, and I use this, many of the books mentioned it, most of the problem in the United States, for instance, we had great sanitary procedures, but those did not really control the polio virus because, as you remember, it's resistant to high ranges of pH and also minor treatments for sewage and so forth. And so if people didn't wash their hands, and if they weren't feeling well, stay at home, there was a natural transmission. Okay, poliovirus is mild clinically in infants and young children, while in adolescents and young adults it may lead to paralytic polio. Paralytic polio emerged as a major health problem due to the development of modern sanitary procedures, sanitation. Before modern sanitation, as I mentioned earlier, nearly everyone was infected as an infant or child, developing a mild infection, primarily due to the presence of passive antibodies to the virus transferred from their mothers. When infected, the passive antibodies prevented the secondary infection and allowed the host to develop antibodies. That's key. Okay, poliovirus infection from a pathogenesis point of view, asymptomatic 90% of the people. Abortive poliomyelitis occurs in about 5% with a minor illness, probably stay at home or go to the hospital. And then there's a non-poliomyelitis or aseptic meningitis, non-paralytic, in 1 to 2%. So you can see a small, and of course the one we really recognize from the poster that started off this lecture, the paralytic polio, bulbar, and which occurs loss of the diaphragm, the ability to breathe. So you go with the iron lung, and you find 75% of the people wind up dying early on. Type 1 polio is responsible for 85% of paralytic polio. That was also important. Type 2 and type 3 are minor, but are not a minor problem, so you do have to take care of them. So that's sort of the pathogenesis. Now, the conquest. This is where I'm going to sort of try and give you sort of a history of what went on with the polio vaccine trials. I won't talk about the 35-36 one that was a fiasco, but the one subsequently. So this is some of the data that looked over many years in actually the United States, looking at and I call your attention to, this is the reported cases, and we're talking about 8,000. So one of the things I want to mention, you'll see it later, is polio is, from one perspective, is not a widespread disease like heart disease or cancer had been. But it was one that, with the advertisements that went out, with kids on crutches, children and adults in iron lungs, and the president, Franklin Roosevelt, being unable to get around, was one that really knocked everyone. And it was one that, probably a good one to select, because with all that basic information that had been generated, we were able to move forward. And this was taken, I think it was somewhere in the Midwest. And this happened in that epidemic in 1916. Many hospitals refused. There's a story of someone whose son came down with what he thought was polio, and he went to a half a dozen hospitals in the New York area, Manhattan and in Brooklyn, and no one was willing to admit him. And I believe, as the story ended, the child died in the car, okay, because no one wanted to deal with it, because they didn't know what was going on at the time. And really they did. It was an infectious disease from 1908 from Landsteiner's work. Okay. This will give you an idea of the average number of cases in the United States in 51 to 55 and what happened after the Salk polio vaccine. So you're talking about 30 to 40,000 cases. And you're talking between 5 and 10,000 people actually dying of the bulbar respiratory poliomyelitis, and also serious infections. So that was the death, and you can see some of the other countries in the world, their incidence was much less, and maybe that's because their sanitation procedures weren't as good as ours. Now, we'll skip that, and so the Letterly Laboratory started the first work on polio vaccine. And actually, it was also shown in 1951, and sort of what the Rockefeller was doing, and that was the use of gamma globulin for treatment of meningitis. If you could give gamma globulin during the early phases of the disease, you could prevent the viremia. The only trouble is the gamma globulin was horse generated, and you could then also react against that. And getting the children diagnosed and into the hospital to begin this therapy was very difficult. So they began to think about vaccine. And obviously Jonas Salk in 1952 was working based on stuff that the Enders group, Weller and Robbins had done in growing cells, of growing the virus and then killing it. And then in 1954 there was a field trial. In 1955 the Salk polio vaccine was licensed, and we'll talk about that in a little bit. And then at the same time, Kaprowski and Cox and Sabin were working on the live polio vaccine. Now, the live polio vaccine is you're giving an attenuated live virus that will infect and replicate in the GI tract and produce antibody, and will produce significant amounts of virus that are secreted into the environment, which can be an issue and a problem. It can also help to develop herd immunity, okay? And there were a number of field trials that occurred, and his vaccine was licensed in 1961-62. This is also taken from the Murray, which is the number of cases that were occurring per 100,000 population, and this is data from the United States, and this is looking at the paralytic only, and this is looking at paralytic and non-paralytic, or total polio. And you can see 41, 42, it's gone up and down. And then when the killed vaccine was initiated, it dropped down significantly. When the oral, it's actually down in, you actually report it now, and we'll talk about why in a minute. This is Basil O'Connor, and this, of course, is Franklin Delano Roosevelt. And I think last year I mentioned that probably back in 1934-35, these birthday balls began, where people sent in money to celebrate the birthday of Franklin Delano Roosevelt, and this sort of was the start of the March of Dimes, or the infantile paralysis. And we're going to talk about that in a minute. These individuals, Basil O'Connor was in charge of it. He was a lawyer and a good friend of Franklin Roosevelt. Turns out that his daughter was also stricken with paralytic polio, but she recovered mostly and was able to be a mother and take care of the kids and so forth. So he had an issue with it too. These are the three individuals that were, not because he made a major contribution, Hilary Koprowski, but I also know him very well and was at the Wistar when he was doing some of his field trials, which we'll mention in a minute. Never met Salk, and I did meet Albert Sabin. But these three individuals made major contributions to development of the vaccine. Salk, of course, gets recognition, as does Albert Sabin and Hillary, if you read the history books. And I recommend, if you're interested, to really read it, because it's very interesting to see the play on individuals as they develop the vaccine in their work. This is Harold Cox, and he was the head of the virology lab at the Letterly, and Letterly played a very major role because a lot of their data went to developing this vaccine. This is basically the advantages, disadvantages of the live oral, and this is the inactivated. The major negative one for the inactivated is you don't get that secretory antibody, so it's not really total protection. And you do have to give booster shots, but it's a lot safer. Well, in a way, it's a lot safer because the vaccine, the virus is killed. Okay? The oral polio vaccine is an issue from one perspective, in that you do have live virus and its potential to revert to a more virulent form. And because you secrete large quantities of it, you can infect individuals. There's a positive and a negative for that. We'll mention that in a minute. Okay, this is a paper, Dr. Koprowski was invited 50 years after the Salk vaccine was out to give a result. He was the only living survivor of the triad. And so what he did was, and I kind of like this, he put together, if you're interested, you can go back and look at it, when all this started. And clearly the Letterly group back in 1950-51. In fact, he created quite a stir in 51. There was a meeting in Hershey, Pennsylvania, where all Enders, Sabin, Salk, Rivers were attending the meeting, and Koprowski was asked to present the stuff from Letterly, and it was an after-lunch meeting, so everybody was kind of, Hershey's a very good place to go, not only to get chocolate, but they do have good food there. Being a Pennsylvanian, I can talk about that. And he woke them up because he did a field trial that hadn't been done since the 1935-36 one that failed by Colmer and Brody. And he actually immunized some children in a mental institution in New York City. And of course, they were created a great uproar, maybe violating the individuals as far as consent, whether they had consented to this or not. But the director of the institute, who I actually met, wanted this done because he was very worried about these children developing paralytic polio to make their diseases even worse. So this was a start, and suggesting that you could develop antibody as a result of immunization, and they were immunizing with live polio virus. This then is a description of the field trials that went on through the 50s, and then the acceptance of the Sabin vaccine. 1955 is when, 54 is when most of the trials went on, and basically showed that antibody was produced and basically ablation of the infection. And I think we can go through here. I want to mention, this is a sum result of the number of people that basically received the oral vaccine by Sabin and also by Koprowski, and there's about 11 or 12 million in both cases. Why the Sabin vaccine was selected instead of the Koprowski was probably for one very good reason. It turned out neither type 1, type 2, and type 3 was any better. Both of them had some issues in monkeys when they were tested, but they were somewhat equivalent. Sabin chose to do his studies in Russia, and if you remember, that's back in the 60s at the height of the Cold War. And the Russians totally moved into the Sabin oral vaccine. So a lot of people feel that politically the United States also had to. So they also selected the oral vaccine of Sabin. Whether that's true or not, a number of the authors in the textbooks that I'll mention in a minute also mention that. And I kind of like this quote at the end of the paper from Kaprowski, because clearly he was, and Letterly and Harold Cox, the first to really move into the oral polio. People wanted to stay away from it because of the fiasco that occurred in 1935 and 36. And he said that in adopting new ideas, the first is it's impossible. Okay, heard that many times. The second is maybe it's possible, but it's weak and uninteresting. Okay, the third is it's true, and I told you so. And the fourth is I thought of it first. Okay, and I think he probably put that in. I've never really asked him. Maybe the next time I see him, I will. I think this is to say that, well, that's okay. But then back in 1966, there's a very good book, The Saga of Jonas Salk, written by Carter, about polio vaccine and breakthrough. And this is a quote from the book. It talks about Hilary Kaprowski, who developed a live vaccine and fed it to human beings with excellent success before anyone else, and had a foot in the field, was aggressive campaigner as Albert Sabin, and ever so much more colorful. He fought tooth and nail for his vaccine, but lost out to Sabin by inches. But now he says he was glad. I consider myself luckiest. He smiles. I'm able to retain my equilibrium. I have become neither a hero as Salk, nor did I become a potentate like Albert. When Dr. Sabin was in Brazil, the children sang beneath his hotel window. I would not like to be in such a position. Few scientists would. Even though he was bitterly disappointed, and I know he was, because I was at the Institute shortly after that was accepted, and there was a lot of quiet down in the poliovirus group. Jonas Salk, and I think it's important to mention this. Both Albert Sabin and Hilary Kaprowski were named members of the National Academy. No Nobel Prize was given for the polio work, okay? Many of the, except for the work of Enders, Weller, and Robbins, who showed that you could grow polio in cell culture. And many of the people felt that that was the recognition for the polio virus, and everything that then followed was simply a matter of developing the technology. And Salk, why we have an institute named for Salk out in California, many people gave money for it. The scientific community basically shunned away from him because they felt most of his development was the people in his laboratory, which a lot of animosity developed because he took credit for everything. And it's, if you read any of these books, it's mentioned in there. He took total credit for it. And it wasn't until the very end, and when he went back for a reunion at the University of Pittsburgh, which is where he did most of this work, did he sort of try to mend the roads, but most of them would not be involved. And the scientific community, while they recognized it as a major important, they didn't felt that he was really the scientist. He was more like a technician. And that's not to denigrate him, because he went and he did some very, very important things by then recruiting some very stellar people to the Salk Institute, which still continues a lot of his work in medical research. Now, with anything new, things result from them. Okay, so first of all, the polio vaccine was grown on monkey kidney cells. Well, one of the main problems with monkey kidney cells is not only do they contain the polio virus that you put in, but they contain monkey viruses. And I happen to work with one of them called SV40, which causes tumors in hamsters and many other animals. And there's some people that think it also caused cancer in humans, although that evidence isn't very strong. So when you get a dose of polio-killed virus, you also get all of these simian viruses. So now, in order for a virus vaccine to be released, the polio vaccine that's grown on monkey cells, they have to be shown not to contain any monkey viruses. And there are ways that you can screen for that. So that, now, in third, Cutter incident. There's a whole book been written, and I'll show you that, by Paul Offit, who's a pediatric infectious disease virologist at Children's Hospital. A company in California, the Cutter Company, produced vaccine which had a lot of live virus in it. So not only did it produce immunity, but many of the children and individuals that received it came down with paralytic polio, and a number of them died. And that was because they didn't pay attention to their inactivation. Whenever you do inactivation here, which is heat treatment and formaldehyde, as I mentioned, it doesn't touch the DNA viruses like SV40. It will inactivate the RNA virus. And Salk's major contribution was determining what the conditions were to inactivate or kill that virus. And those had to be stringently adhered to and had to be biologically tested. And so the killed vaccine, which it shouldn't have, actually produced polio. That's from the cutter. The issue, and that's number 5, the issue with the oral vaccine is you get reactivation of the virulent virus, okay? And this can be an issue, and from the World Health Organization point of view, it is a major issue, let's say a minor issue, but with major consequences, as we'll see in a minute. Number 6, this is an interesting one, and for those of you that want to follow it, it's very good. There's a whole book that's been written about it called The River. An article came out in Rolling Stone that basically made an accusation that the oral poliovirus trials in the Belgian Congo was how AIDS started by passing of this monkey virus to humans through chimpanzees. So they made a claim that the Belgian Congo trials, which was the Lederle, the Wistar Institute, Hillary Koprowski, actually transmitted HIV. So for about 5 years, this was a major, major issue, and this whole book has been written about it. The good news was that an international panel was put together, and first of all, they found out that the original vaccine was still available, and they were able to do DNA sequencing studies and found no chimpanzee DNA, only African green monkey DNA, and they found no evidence of HIV in the vaccine. So the Koprowski group has in a sense been exonerated, but there's a whole book that's been written on it, and another very major. Now, cost analysis, and I think this is one that is kind of interesting from the perspective of does research pay for itself? Okay, so there's an article, and I draw your attention to it, called Retrospective Cost Effectiveness Analysis for Polio Vaccination in the United States by Thompson and Tebbins. And it's in the journal Risk Analysis, volume 26 in 2006. And they went back and they gauge everything to 2002 figures. And so this is the following information, some of which I put together. The cost for vaccination from 1955, the Salk and the Sabin, turns out to be about $35 billion. Okay? And they also claim that it's going to be about $1.4 billion from 06 to 2015. And they also have in the paper, these are Harvard authors from public health and also MIT, and they have a very elegant mathematical formula at the back that actually looks at all of this in the various categories. So the total cost of the vaccination in the United States has been about $37 billion. And it's prevented because you can take those figures every year and total them up from 1955 as to how many people would come down with poliomyelitis, how many would develop paralytic, how many would die. And these are the figures. There would have been 160,000 deaths and 1.1 million cases of paralytic polio. Based on that calculation, it's basically saved $180 billion in the United States alone by prohibiting that 160,000 deaths and 1.1 million. And this doesn't include the mental anxiety amongst the families and all the long-term care. Probably they calculate it's really more like $800 billion. Okay? Now, what is the investment in research? Now, this is kind of difficult for me to pull out, but I can do it. I can't figure out how much money the Lederle put in. That's proprietary information, but I can make a guess. It's probably not more than $50 or $100 million, even with the clinical trials back at that time. But the investment in research from Landsteiner's point of view and from Simon Flexner is probably some dollar figure less than $400 or $500 million over about 50 or 100 years. I mean, it doesn't even approach $1 billion, but we can make that. The March of Dimes spent $70 million on research, most of which funded Jonas Salk's development of the killed polio vaccine, and a significant portion also funded the work of Albert Sabin and also some of the other virologists that I mentioned, like John Ender's lab, the Ender's lab, and also the Rivers lab, and other polio investigators. The NIH, which was just starting back in the 30s and 40s, we only spent $7 million from the NIH. So that's the total amount of figure plus whatever Letterly sent. And my guess is it's way under probably $500 million or $1 billion in research cost. So not a heck of a lot. And I think if you go back in time and calculate the total cost that it saved, and also when you go through nationally, it's quite a lot. But I do direct your attention, if you're really interested, to look at this article. They do a fantastic job. This is also from their article. This is the number of polio cases, and it sort of has its ups and downs, and there had a peak over here. You're talking 20, 30,000 cases, and then there's a break, and that's a result of the Salk polio vaccine here, and now we're talking 20, 40, 60, 80. You can see it coming down, and then it's 0. Now these little bars here are cases as a result of the vaccine. That's like the Cutter incident, okay? Because sometimes the inactivation procedure, although I think our controls are much more stringent now. And then of course we also then developed the oral polio vaccine. So a lot of people have gotten both. I know I have. One of the authors, actually, Oshinsky, and I kind of like his book. It's a more recent one. And he talks about, as I'll mention later, the personalities that were sort of involved at the time. And he made a calculation as to the amount of money that was raised, and actually for infantile paralysis research back in the 50s. And he says the foundation raised about $66 million. And then we look at the other major diseases, TB, cancer, cardiovascular, cerebral palsy, and he also took the data from 1954 as the number of cases. And you can see that polio is one that's the least, but it's one that I think the advertisement and the fact that you mobilize the community, Franklin Roosevelt, and also the acting community, actresses, and had these fundraisers throughout the country. And basically the people were the one, even though our money also goes to NIH. The March of Dimes did a significant amount of research. So the March of Dimes raised between 38 and 62 about $630 million. 70 million was allocated to research. The rest of it went to take care of the patients, the iron lungs, as well as the people that developed paralytic polio. And of course the 70 million was 10x the amount that the NIH used. This is just a sort of a little description of some of the business about starting the birthday balls. The first 1.3 million that was raised, and people were sending in dimes that were coming into the national office. Basil O'Connor was made the head of it as far as organizing. And as I mentioned last year, Paul de Kroof was the original scientific advisor, but he made some very major mistakes, although he got them on to thinking about research as a potential way to eliminate this. And this is just a quote from O'Connor, being the par element of a lawyer. He just couldn't understand. He understood money and power and didn't understand scientists. And I bring this up again because I kind of like it. O'Connor didn't understand scientists. As a powerful lawyer and advisor to the president, he was used to getting, he wrote many of his speeches, by the way, used to getting what he wanted and getting it quickly. O'Connor understood men who wanted money, and he understood men who wanted power. But scientists, apparently moved by neither, were harder to compel to do his bidding. Remember, his bidding. They all seemed perfectly comfortable, huddled in their laboratory, studying one small aspect of one small virus for the rest of their lives. They weren't in any rush to do anything. O'Connor was frustrated by their ponderous, circumspect, measured ways. But I think if you listen to what I've just said about the history of polio, leave it up to the scientists, not to the businessmen. Okay? Because we really need to understand, as you'll see my plug at the end, before we move forward. Now, where are we with polio now? As you can see, Canada and the United States are completely wiped out. This is 1988. These were the endemic areas. As in 2006, it's been eliminated because the World Health Organization has taken the oral as well as the kill vaccine. Most of it's oral worldwide. And there's only 4 endemic countries as of 2006. And I haven't checked recently, the World Health Organization. It's probably much reduced. And in fact, now what we're reporting are these outbreaks as a result of the virus that's being reverted as a result of replication, the oral polio vaccine. So there have been a number of major outbreaks in the Dominican Republic and Haiti. 2000, there were 22 cases that could actually be traced back to the oral vaccine. They were sequenced and they were the same. So we've got to keep an eye out for this, and you can see there have been a number of other cases. But the good news is those, just from the United States alone, when you're talking about 20,000 cases, you're talking under probably 100 to 80 cases worldwide now. So this has been a major, major success. Readings and references. Okay. This is a book from 1960, and of course this is at the time when everything happened, and for as far as the polio, as far as Enders, as far as Sabin, Salk, and Kaprowski, and it's extremely interesting reading. So when I had decided to do this, I pulled it out of my library and had to dust it off a little bit, and really enjoyed reading it again. This is another one, and I took a quote from it on Jonas Salk, and there's some, and I lifted a couple of quotes out of here. Carter actually spent some time interviewing, because at this time I believe in 1966, all of the individuals that were involved in it were still around. John Paul, I already mentioned some of the figures I took from this. This is really, I would say, a masterful treatise. It's more like an encyclopedia. It goes into great detail, and it talks about everyone, and that's very good. It's not only talking about the dominant figures, but everyone that contributed. So there were people there that I knew of that I didn't know actually worked on polio, and they also made major contributions. This is a little one that came out in '70, Aaron Klein, and I think he did a very nice job. He again went around and interviewed most of the individuals that were involved in the vaccine, and I kind of like his rendition of Dr. Kaprowski and Dr. Sabin and also Dr. Salk did an elegant job. This one is a more recent one, Oshinsky, and it's very well done. He has a lot of information in there about it, and he's been able to go and find out sources elsewhere. This is a very nice one too, A Summer Plague, Polio and its Survivors, and vaccine, talks specifically about all the vaccines, the other ones that I mentioned, the rabies vaccine, and also the Jenner vaccine against smallpox, but devotes quite a bit to the polio. For those interested and want to talk about and find out about the Cutter incident, this is something that really blights science and the pharmaceutical industry. Is you really have to be careful and make sure that you're well controlled as far as what you're releasing to the public. And this could be why vaccines have gotten sort of a very, very negative rep. When I just went and looked at vaccines and polio vaccine on the internet before I came over, there's still a lot of articles on there about SV40 and about simian viruses. So we really do have to know, and we have to be well controlled. One of the Nobel Prize winners, the Enders Group, Thomas Weller, actually wrote this book. It's about 5 years old, and it really gives a personal insight to a pediatric infectious disease person working in a very good virology lab, and being allowed to do what he did and made major, major contributions not only to polio, but also to other infectious disease. And then if you're interested, Hillary wrote a book. I'm not totally enamored with it. Hopefully he doesn't hear that. But it talks about some of his entree-vous into science, not only polio vaccine. And this is that book that talks about the Belgian Congo trials and the potential transmission of HIV as a result of the potential use of chimpanzee cells for the growth of the polio virus. And this is a more recent one that came out, and our library has it, Jeffrey Klunger on the Splendid Solution. It's about Jonas Salk. Now, the conclusions I'd like to make: research creates knowledge, and I think the polio story is a great example of that. It doesn't happen overnight. I mean, polio has been with us since the Egyptian times. This is 1500 BC that tablet dates back from, okay? But it's really been over the last probably since about 1950, 1940. So it's been basically a half a century, and really less than that, that we've been able to make this major inroads. It develops an understanding. It leads to prevention and cure. It's very cost effective, and that's why I did the cost analysis, because these were figures that stayed with me. So every dollar we spend in research, and there's some kind of interesting stories, and that was a story in the Oshinsky book of a lady, and I can't remember what city she was in, just a month before the vaccine trial started. Her daughter came down with bulbar poliomyelitis, and at least the way it was written, she probably didn't survive 2 weeks as a result of the major nerve damage, okay? And of course, the quote in the book, because I think he interviewed her, was that why couldn't she have been, administered the vaccine? Why couldn't the vaccine have been out sooner than later, okay? So the human suffering, which we haven't put a figure on, but probably approaches that 800 billion or trillion. By the way, that's our debt, isn't it? Okay? So research, I think, really does a great job. Okay. The end. Research saves lives, and I think it also is very cost effective. And considering healthcare cost or reimbursement going down, that's very important. Thank you.
So, does anybody have a question?
On the post-paralytic, or is it post, the sequelae or the later, is that from being vaccinated? Or what's...
The post-paralytic syndrome?
Yeah.
Okay. I didn't mention that. It's on one of the slides. Okay. I talked about the acute phase, not the chronic. It turns out that most polio victims, and I actually know two very well. One of my executive assistant at Albany, Joanne, walked on crutches, and the chair of public health at the University of Colorado, Carlos Martini, also had paralytic polio as a child before the vaccine. And both of them both worried about, as they hit their 50s, 60s, and 70s, what happens is you get reactivation. So the nervous system is able to make some reconnections, but then at later, like 30 and 40 years after, they begin to fail. And as a result, they get worse and worse from the point of view of not being able to move around and get around. So this is a major problem, and actually there's a lot of healthcare costs going into that because these patients need attention, they need rehab, they need treatment. And I can tell you, everyone that I know that's had paralytic polio or suffered from a withered leg like that Egyptian priest realizes that they will have this issue. And it's a very, very major medical problem now. It's not good news. The news is the number of patients are much reduced than what we've had before. Okay. So that's an area I did not spend any time on.
Do the individuals just have to know that they had polio and be aware that later on they can have these?
Yes.
Or
I
if
think
you
they're
don't really think about that, does it like go undiagnosed as really a post-paralytic polio situation?
No,
or
those
is
individuals that will develop this
Yeah.
post-paralytic know that they had, and they have paralysis in some of the limbs and other issues too. Okay, so you generally know it, yeah.
I could just see multiple kinds of testing and cost
Yeah.
if they really didn't know that up front, trying to figure out
Oh, okay.
I was just thinking that. So,
Okay.
yeah.
Anyone else?
Can you comment on how you see the eradication of polio? It's limited down now to 4 countries, but there's a lot of skepticism in terms of whether polio can be eradicated. I guess the strains 1 and 3 are still prevalent in areas like Afghanistan, Pakistan,
Yeah.
India, areas where there's some skepticism that we might get a final eradication, global eradication of polio. How do you see the future?
I think the key is going to be getting the vaccine out to these individuals and getting them vaccinated. Okay, that's one. Number 2, we also have to understand something about the reactivation of the virus, because we're getting a small number of infections as a result of virulent virus coming back again. But I think the key is, and what I've read about the World Health Organization, they're trying to get it out to everyone. And obviously, the ease of giving the live oral vaccine seems to be the method of choice. Put it on a sugar cube and give it, and then it's done, as opposed to the Salk. In this country, we still use the Salk because of this potential for reactivation. Some of us have gotten, I don't know how many immunization shots I've gotten. I've got 5 or 6 Salks, and I've taken, I don't know, a dozen sugar cubes because I work right down the hall from a major polio lab. So I would assume my antibody titer is very high. So I think it's a matter of being able to identify and then making sure that you are getting the majority of population. The good news about the oral vaccine is if these people are nomadic and living together, if you can get one immunized, the likelihood is you'll get many more immunized because they'll be secreting the virus and they'll get it as early. And you know, most of them are still, by herd immunity, are maybe going to be transmitting antibody or as a result of passive antibody to the young children. If the young children get infected, then they'll become resistant as a result of producing that primary antibody. I'm thinking that's getting the vaccine out there is the key.
Thanks.
Anyone else? Okay, well, thank you very much, Dr. Lehman.
Okay.